This is a demo of Strandomics, a platform for the interpretation and reporting of rare disease cases. So if, as an interpreter, if I log in, what I see is a set of cases that await my attention here. The first case is a trio case. The remainder are single sample cases. I could look at the individual samples by clicking on them, and that opens up the test request information as to when the sample came in, various details of where it came in from, date of collection, etc., etc. But most importantly, the clinical manifestation of the sample. This is a male child born to consanguineous parents, prominent eyes, respiratory problems, sudden death, systemic and pulmonary hypertension, pyloric stenosis, etc. So that's the phenotype of the case. And linked to this sample, of course, are the two parental samples. They're all linked in the system. And if you click on the parental samples, you'll be able to see which one is the mother and which one is the father. So going back to our case, let's open this trio case. And each case, as you can see, has a set of roles associated with this case in order to get the report out. At Strand, we have a role called interpretation, which is the first pass of looking through the variants, annotating them, and creating the report, which is followed by a reviewer who reviews the report that's created. And then there are QC steps and report sign-off steps, and these are all separate individuals who eventually participate in the report creation process. There are also other roles like observer roles, which is useful in a training setting, etc. So this is the case. So let's open the case, and now when we open the case, we're going to see all of the variants associated with this case clubbed at the gene level. When the VCF file is imported for this case, or the three VCF files, one for each of the individuals, is imported, then all the variants are assessed for pathogenicity using a collection of rules, and labels are assigned to each of the variants. And this is done automatically at ingestion time, and once you open the case, all of those labels are available. And there are a number of genes containing variants. As you can see, there are 8,390 of these genes. This is a whole exome case, which I perhaps forgot to mention earlier. So this is a large number of variants, and not all of them are going to be important. Of course, this is sorted by pathogenicity, but as you can see, there are a large number of likely pathogenic variants. Not all of these are likely to be important in the case at hand because of the genes, associated genes may not have any phenotypes, etc. So you would typically see some pictorial symbols here in instances where the gene is associated with some phenotype. Now the first step, of course, would be perhaps to filter this long list down by phenotype of interest. So one could enter one or multiple phenotypes. So for example, pyloric stenosis, and that automatically filters the list down to 181 genes carrying variants. One could also set the appropriate inheritance hypothesis. In this case, the parents were unaffected, so we choose the recessive hypothesis, and that leads to 181 genes, and not all of them have been automatically assessed to have a substantially high label, so we could just pick those which have been automatically assessed to be a VUS or pathogenic or likely pathogenic. And as you can see, we actually have multiple subcategories within the VUS based on, again, a collection of rules. And typically it's the higher categories that are important, and of course if one doesn't find anything amongst the higher categories, our interpreters would continue to look further and further down. So let's say we pick these, and so this is the list of genes that we have at hand. And you can see there's the filamin A gene here, the fibrillin 5 gene, and a few other genes. In front of these genes is a red mark which indicates that these variants are perhaps of poor quality, and that leaves only these two variants which are of reasonable quality, green and orange. So the filamin A gene is involved in a large number of diseases, and those are mentioned here, and the associated organ systems that this gene hits are given here. And out here are in black the variants in this case, and in blue are the variants generally known in dbSNP, ClinVar, etc. laid out. This is just to see whether variants that are well known occur in regions where the case variants occur as well, though this view is not commonly used by our interpreters. Now, in order to assess each of these variants, one would ideally like to drill down into the gene and consider the variant information itself. So, for this gene, you have a variant that's an aspartic acid to asparagine change. Eventually, if you want to report by transcript, you can pick which transcript to. We have done some work to ensure that the most common transcript or the canonical transcript is the default transcript that's used. Now, this variant is, as I said, aspartic acid replaced by asparagine. It's predicted damaging by 6 out of 7 predictors. It causes a change in amino acid properties from a negative charge to a neutral charge, so that's important. The amino acid is conserved in primates, vertebrates, and mammals, as shown in this plot. And then the variant information itself appears here. The supporting read percentages, the number of reads, and the zygosity, all of that appears here. And as you can see, this variant, the proband, the child is hemizygous, and the mother, female, the mother is heterozygous. The father, of course, does not have the variant. It's an X-linked variant, so the father has no role in passing the X chromosome onto the child. One could also see the reference sequence and the actual change out here. Here are shown whether the variant is present in various population databases. If it were present in exact 1000 genomes, etc., dbSNP, it would show here, and clearly it's not present, so nothing shows here. Here, if the variant is present in our internal curated database, that will show here. And here it would show if it's present in ClinVar and associated databases. So this is the pooled patient database, which is the database of all of the patients that whose data has passed through the Strandomics system. So automatically every variant will be looked up against all the previous patients. So in this case, this variant is present in only 2 out of the previous cases. Now this is a demo server, so there are only 48 cases, but on our real production server, this will run into perhaps 10,000 or so. And there are only 2 cases here, and one could drill down which 2 those are. Of course, those are likely to be just these 2. So one could click on this and see which 2 cases they are. Both of these are whole exome cases, and one of those is the proband at hand, and the other is the mother. And in general, of course, if there were other previous samples with this variant, the details of those samples, including the phenotypes, would all be available for assessment. So after looking at all of this information, one could also check the gene information to see which diseases the gene is involved in, and what is the typical inheritance mode. And then there's a lot of information around the phenotypes of each of these diseases. So one could look through all of that to make a decision on this gene. One could also look at the transcript view, which carries important technical information about the variant. For example, this is the variant at hand, and you can see in each of the 3 samples the coverage around that variant. So let's drill into this region, and here the coverage is 30 in one sample, 20 in another sample, and 50 in another sample. So you can see that in all of the samples there are sufficiently many reads. And if we drill down a little further, we'll see that whichever variants are known in dbSNP or in other databases like ClinVar, Emory, OMIM, etc., none of those match the variant at hand. So it's a completely novel variant. The gene structure is shown here. And finally, also shown are all the binding partners and where they bind to the gene and the associated domains of the gene. You see our variant is here and it's present in an area where there are lots of proteins binding. With all of this information, one can make a decision on the variant at hand, and the way one makes the decision is to check this checkbox, which will ask for the entry of some notes, and these notes are kept. These are informal notes, but you can keep adding notes as you go along to retain information, and these are always time stamped, and the entire audit trail of all the notes is kept as you go along. If needed, one could also take this variant and send it for lab validation right here, and then it'll a separate list will appear. If you have a role as a lab validator role and you log in, you'll see a list of requests against your name. You can also submit a curation request where you publish information about the variant, and all of this information, and you could enter extra information, will get published into the curated database and be available for all future variants. Okay. Now, having selected the variant, one could then select, also look at the gene itself and add notes on the gene on whether it matches the phenotypes. So for example, here we say FLNA has wide-ranging implications, and there's a chance that this variant could be causative of the phenotype because there's a really wide range of phenotypes that it has, but there is no direct evidence, and so on. So you could keep notes and check genes that are interesting. So all of these checks and the notes are all kept as audit trails for the reviewer to come in and review what's really happened. And then once you pick whichever variants are of interest to report, then you can go into the reporting mode. But before that, let's illustrate some more filtering options that exist. So one could also set various gene lists to filter. In Strandomics, there's a large collection of gene lists that we have curated for different sorts of diseases. So these are all curated gene lists by our curators, and you can see the curator name. Each of these lists, and there are many such lists. So one could take each of these lists. One could take out here, apply whichever list one wants. So you could search by gene list and say I want to apply a particular gene list, and pick that gene list and apply it, and that would again filter the list of genes available in this dashboard down to the appropriate set. So that's yet another way of filtering, in addition to the filtering based on HPO phenotypic terms. And then there are other ways of filtering as well. So you can preset filters. For example, if I set this filter, it will show you what it does. So you can set preset filters of various kinds. We haven't allowed for a very dynamic setting of filters right here, because that will make the production process of interpreting a large number of tests very free form. So instead, we set up a collection of filters beforehand, and then the interpreters can set whichever filter they want on the fly. Typically, the SOP involves setting these filters in turn. The first filter would be the most stringent, and the second would be less so, etc., so that you can work your way through in decreasing levels of stringency. Having said that, now we picked the variants we need to, and we can go into the reporting part. So when you go into the report, the variants which have been selected are shown, and you then get to assign a new label. Now this is the manual step of assigning the label, and in this step, all of the groundwork that's been done earlier is used to assign a label manually. We do have SOPs and tools separately for humans to for the interpreters to decide which label to assign, and at some point of time we will integrate those tools into this platform itself. But as of now, this manual label overrides the automatic label. And once you assign this, you can proceed to the report, and that report essentially copies over all of the variants and the notes that you had added earlier, and based on which you can now fill in a few text boxes, including the gene summary and the variant summaries, etc. And once you fill those in, and if this information had been already filled in for a previous variant that was seen earlier, then that information will be automatically copied. As you can see here, it's already present. If you want to add references and citations in the text, all you need to do is to include the PubMed ID in double brackets, and automatically when you generate the report and get to the sign off stage, the report is automatically generated. It includes the variant information, including the HGVS nomenclature and all of that copied over automatically, and then all of the information that was entered during reporting time formatted out, including citations, which are automatically expanded here. And then templates can be set to add all of the other supplementary information on, which genes, which methodology, all the standard templates that go into the report. In addition, it's important to note that each of these boxes can also, we don't want interpreters to be entering free from every time, so we've created a number of templates by which these can be auto-populated. So for example, for every type of variant, we've got templates. So missense variants, when you click on it, you'll see a template of this kind with parameters where the text is just automatically added in with the appropriate parameters filled in automatically. And there are a very large number of templates, both for the various top types of variants and the summaries, and various sorts of recommendations based on different tests. As you can see, there's a lot of templatized text that's been created already. Okay. Now that's pretty much a rough overview of the entire process. Also note that if there's a variant that is not present in the VCF file, but you do some extra analysis outside and you want to bring it in later, there's a way to inject variants. So these injected variants are automatically shown here and also can be added to reports. So in particular, for copy number variants, structural variants, process involves, at Strand, the standard process involves not putting them into the original VCF file, but taking them through a separate analytical process. We have a collection of tools around that. And then whichever variants are selected through that process are then injected in here. And as you inject them in, automatically the HGVS nomenclatures, etc. are assigned, and then they are carried through into the report. So that is should give you a good feel for the entire Strandomics platform. You should also note that the Strandomics platform is very elaborate and has been used for, continues to be used for several hundred reports every month and going back for now several years. So perhaps more than 10 or 15,000 reports have been generated using this platform, and it's used both as an interpretation platform as well as a workflow management tool in our lab. And so you'll see all associated functions, all the way from managing cases, configurations, curation requests, to payments, counselor activities, SOPs, setting up tests, setting up templates to report variants, etc., gene lists. All of that is present in the system, and we can go into more detail if the need so arises. But hopefully this will give you a good feel for what the platform is about. Thank you very much.